Hello everyone, welcome to a discussion of important topic for first MBBS examination, also for the paramedical students, that is digestion and absorption of lipids. According to the MCI competency, it is included in the competency 4.2, which deals about the digestion and absorption of lipids and disorders. From this topic, you can get BAQ, that is role of bile salt in the digestion and absorption of lipid. Second BAQ, that is brief answer question, is various types of lipases. Okay, and SAQ itself, that is digestion and absorption of lipids. Now see, these are the key words while writing the answer. First one is the emulsification, then the name of bile salt and role of bile salt in the digestion and absorption. Pancreatic lipase, which is present in the intestine, and action of the pancreatic lipase, it is important. Mixed micelle formation, which is very important for absorption of lipids into the intestinal cell. The process is known as simple diffusion. This absorption process is known as simple diffusion. And what is role of chylomicron here? So this is very important and key words in the digestion and absorption of lipids. So let's discuss. So what do you mean by emulsification? Emulsification means, the meaning is dispersion of larger lipid particles, dispersion of larger lipid particles into smaller ones, dispersion into smaller particles, that is known as emulsification. And what are the requirement for the emulsification? So bile salt is required, this is the first. So for the emulsification, bile salt, it is required. Bile salt reduces the surface tension and it causes the dispersion of larger lipid molecules into the smaller ones. Okay. Second, then amphipathic lipids, mainly the monoacyl glycerol, phospholipid, diacyl glycerol, these are required. They act as a surfactant. Okay. And the third thing is the peristaltic mixing. Just wait. Peristalsis is important, mechanical mixing in the intestine. So these three things are important for the emulsification of lipid, means larger lipid droplets. Because of bile salts, these larger lipid droplets, they are converted into smaller one because of reduction of the surface tension by the bile salt, detergent action of the bile salt, and the these phospholipid, monoacyl and diacyl glycerol also help in the emulsification process, and the peristaltic mixing. This is important for the emulsification. Now, what is bile salt? What is the role of bile salt here? So, can you enumerate some bile salts? So these are the glycocolate. Remember this, glycocolate, taurocolate, sodium and potassium salts of glycocolate and taurocolate. Now remember, the cholic acid and chenodeoxycholic acid, and chenodeoxycholic acid, these are the bile acids. Now their conjugation occurs by the glycine and taurine, so they are converted into glycocolate and taurocolate, glycocenodeoxycholate and taurochenodeoxycholate. Now sodium and potassium salts of these glycocolate, taurocolate, glycocenodeoxycholate and taurochenodeoxycholate, these are known as the bile salts. Bile salts, sodium and potassium salts of glycocolate, taurocolate, then glycocenodeoxycholate and taurochenodeoxycholate. These are the bile salts. Now what is the role of bile salt? It reduces the surface tension and larger lipid particles, they are broken down into the smaller particles. So what is this action? Why this action is required? Why these larger lipid particles, they are converted into smaller ones? This is known as dispersion into smaller droplets, that is emulsification. Now because of this reduced surface tension by the bile salt and amphipathic lipid, these smaller particles, they have the more surface area So more surface area is available for the action of pancreatic lipase via this process dispersion, because these are the lipids, means these are the water insoluble compounds. So enzymes not act directly on these water insoluble compounds. So larger surface area is required, and this is provided by dispersion of lipids, larger particle into the smaller ones. So three things are required for the emulsification. The first, that is bile salt, then amphipathic lipids, and the peristaltic mixing in the intestine. These three things are important for the emulsification. Now what is the role of bile salt? It causes the emulsification. This is the first role, and second role is it is important in the mixed micelle formation that we will discuss. So pancreatic lipase here important, because pancreatic lipase is the main enzyme. Then phospholipase A2 and cholesterol esterase, these are the main enzymes which are secreted into the intestine, and they are responsible for the conversion of triacyl glycerol into the fatty acid and the monoacyl glycerol. Now what do you mean by mixed micelle? That we will discuss. The bile salt at the periphery and the product of digestion. Digested means the complex molecule, they are broken down into simpler one, and these molecules and the bile salt, they together form the mixed micelle, and they are absorbed into the intestinal cell by the simple diffusion process, and they are incorporated into a chylomicron. This is lipoprotein. Now we'll discuss the digestion and absorption. See what we eat in the diet. So what we eat in the diet, that includes the mainly 90%, it is triacyl glycerol. This is the major lipid, triacyl glycerol. Okay, then followed by the cholesterol, cholesterol ester, phospholipids, fatty acid. So these are the compounds what we eat in the diet. Triacyl glycerol contributes more, major component of the diet. TAG, neutral fat, it is also known as neutral fat. So fatty acids, they are attached to the glycerol molecule. So these are the compound what we eat in the diet. Now what happens in the mouth, stomach and intestine? See, in the mouth, the digestion is negligible. Why? Because transit time is very less. Also lipase action is, no lipase action is there in the mouth, because it is secreted by the dorsal surface of the tongue, lingual lipase. So lingual lipase, it is secreted by the dorsal surface of the tongue, and when food reaches the stomach, there are two types of lipase in the stomach now. One is secreted in the mouth, that is lingual lipase, and second is the gastric lipase, which is also known as acid stable lipase. Acid stable lipase. Generally these lipases, means lingual lipase and the gastric lipase, they are active at the neutral pH, and they are particularly active in the infants. They are particularly active in the infants and young children's. Infants and young children, and these infants and young children, they specifically they consume the cow's milk. So cow's milk contain the short chain and medium chain fatty acids. So short chain fatty acid and medium chain fatty acids means the length of hydrocarbon chain less than 12 carbon, less than 12 carbon. So this is present in the cow's milk and the dairy product, that is short chain fatty acid and medium chain fatty acid. So these fatty acids, they are digested, or they are degraded with the help of lingual lipase and acid stable lipase. So they will not convert the long chain fatty acid into free fatty acid. They will not convert the triacyl glycerol, the acyl coenzyme A, the fatty acid chain in the triacyl glycerol, when it is a very long chain fatty acid. So they act on short chain and medium chain fatty acid where length of the hydrocarbon chain is less than 12 carbon. So lingual lipase, acid stable lipase. Now when the chyme, when the food reaches the intestine, when the food bolus reaches the intestine, there is secretion of two important hormones, that is cholecystokinin and secretin. Cholecystokinin and secretin. So this cholecystokinin causes the contraction of gallbladder and releases the bile. Here is the role of bile. It causes the contraction of gallbladder and releases the bile. Also, this acts on the pancreas for release of the pancreas for release of pancreatic enzymes. This secretin, it causes the secretion of bicarbonate. It stimulates the secretion of bicarbonate and neutralize the acidic pH of the chyme. Chyme means food bolus. Food bolus, after the stomach, it reaches the duodenum. When it reaches the duodenum, there is release of bile. Because of the action of cholecystokinin, there is release of pancreatic enzyme, specifically the pancreatic lipase, and secretin release the bicarbonate and neutralize the acidic pH, which is important for the action of pancreatic lipase. So the digestion principally occurs in the intestine. Digestion occurs in the intestine. Now, how the digestion occurs? Now see, triacyl glycerol or triglyceride is the principal component. Now this is, see, CH2, CH2. This is glycerol to this CH2, OCOR1. This is the acyl coenzyme A, fatty acid attached to the glycerol at the C1. Okay, now C2, this is the C2 position. This is R2, and the third one, this is R3. So this is glycerol, this is C2, this is C3, C1, C2, C3 of the glycerol, and this is the structure of triacyl glycerol, and which is a major lipid, and digestion of TAG is important. Now, how TAG, triacyl glycerol, is digested? There is action of pancreatic lipase. Pancreatic lipase will act on this triacyl glycerol, specifically at this C1 and the C3. At the C1 and C3 in stepwise manner. So it is acted upon by the pancreatic lipase, which is the important enzyme in digestion of lipid. Pancreatic lipase will act on C1 and C2 in stepwise manner. Okay, and it causes the formation of this compound. Only the fatty acid at C2 position remains. So it is known as 2-monoacyl glycerol, plus it will give free fatty acid plus free fatty acid. 2-monoacyl glycerol plus free fatty acid plus free fatty acid. This is action of pancreatic lipase. It requires the bicarbonate to neutralize the acidic pH of the stomach. It requires the bile, action of the bile. It requires the emulsification of lipids into smaller droplets, so that the surface area will increase, and when the surface area increase, the pancreatic lipase will act. So this is action. Okay, so after the TAG, the next important is the phospholipids. How phospholipids are digested? See, phospholipids, it is acted upon by the phospholipase A2. Phospholipase A2. Now see, this is structure of phospholipid. This is glycerol. If this is glycerol, this is fatty acid, this is fatty acid, this is first, second, this is phosphate, this is nitrogen base. This is the structure of phospholipid. So phospholipase A2 will act on this fatty acid and will liberate this fatty acid. So it converts the phospholipid into lysophospholipid plus free fatty acid. Converts the phospholipid into lysophospholipid and free fatty acid. So this is second important enzyme, phospholipase A2. It acts on the intestine and on the phospholipid. And the third compound is the cholesterol esters. Cholesterol esters. It is acted upon by cholesterol esterase and convert it into free cholesterol plus free fatty acid, free cholesterol and free fatty acid. Now these are the three important enzymes: pancreatic lipase, phospholipase A2. Pancreatic lipase cleave the C1 and C3 of the triacyl glycerol, convert it into monoacyl glycerol and two molecules of free fatty acid. Phospholipase A2 act on the second carbon of the glycerol, will release the fatty acid attached here as a free fatty acid, and the lysophospholipid will form. And cholesterol ester, it is acted upon by cholesterol esterase and converted into cholesterol and free fatty acid. So these are the compound forms formed in the digestion. So how many compounds are there? See, there is formation of monoacyl glycerol, there is formation of free fatty acids, there is formation of the lysophospholipid, there is formation of cholesterol, there is formation of phospholipids, lysophospholipids. So these compounds are surrounded by the bile salt. They are surrounded by the bile salt. They are surrounded by the bile salt. Monoacyl glycerol, free fatty acid, diacyl glycerol, cholesterol, phospholipid, lysophospholipid. These compounds surrounded by the bile salt. This is known as bile salt at the periphery. So this is known as mixed micelle formation. Mixed micelle formation. So in the mixed micelle, the lipid in the interior and bile salt at the periphery. Lipid in the interior and bile salt at the periphery. That is known as mixed micelle. So see, triacyl glycerol, it is broken down into free fatty acid and the monoacyl glycerol. Cholesterol ester, it is broken down into cholesterol and the free fatty acid. The phospholipid, it is broken down into lysophospholipid and the free fatty acids. So there is formation of free fatty acid, there is formation of monoacyl glycerol, there is formation of cholesterol, there is formation of phospholipid, lysophospholipids. So these compounds along with this bile salt will form the smaller lipid droplet, droplet, smaller than the lipid droplet. It forms the molecule smaller than the lipid droplet, that is known as mixed micelle. And it is important, and this molecule mixed micelle, it is absorbed through the intestinal cell. It is absorbed through the intestinal cell by the process that is known as simple diffusion. The process is known as simple diffusion. These molecules will internalized monoacyl glycerol, diacyl glycerol, lysophospholipids, then cholesterol and free fatty acids. Monoacyl glycerol, diacyl glycerol, lysophospholipid, cholesterol, free fatty acid. These molecules are internalized into the intestinal mucosal cell. Bile salt will it is absorbed later in the terminal part of the intestine. This simple diffusion specifically occurs in the jejunum and ileum part of the small intestine. So this is mixed micelle formation. This is simple diffusion. By the simple process of simple diffusion, it is absorbed. Now after the absorption into the intestinal cell, there is resynthesis or re-esterification occur. Re-esterification. What is the meaning of this word? So there is synthesis of, in the intestinal cell, there is synthesis of triacyl glycerol occur. From this free fatty acid and monoacyl glycerol, they are converted into triacyl glycerol. So there is synthesis of triacyl glycerol occur, there is synthesis of phospholipid, there is synthesis of cholesterol ester. Occur in the small intestine again. Okay, so that's why it is known as re-esterification. So in the re-esterification, there is synthesis of triacyl glycerol, phospholipid and cholesterol ester occur in the intestine. Along with these, through the mixed micelle formation, vitamins all A, D, E, K, fat soluble vitamins, they are also absorbed through this mixed micelle. Now there is synthesis of one protein in the intestine that is known as ApoB48. ApoB48. It is 48 because only 48% is produced in the intestine, while the same mRNA will give the 100% protein in the liver. And this is example of RNA editing, which is an important RNA editing. This is an important MCQ, RNA editing. It is produced in the intestine because of premature truncation of protein synthesis, and only 48% of protein is produced. So intestine, this protein near about, this contribute near about 1% protein in the chylomicron. Okay. So this together, these structures together will form the important structure that is known as chylomicron. So in the chylomicron, see, in the inner core of the chylomicron, there is triacyl glycerol. So this is triacyl glycerol, which is in the inner core of the, this is triacyl glycerol. Then second important compound in the inner core of the, this chylomicron is cholesterol ester. It is cholesterol ester. So this is cholesterol ester. So the neutral lipids means the hydrophobic lipids, non-polar lipids, not neutral, non-polar and hydrophobic lipids, TAG, also known as neutral lipids. So these lipids will be at the inner core in the intestine, in the intestine where chylomicron is produced, and it is surrounded by the phospholipid bilayer. It is surrounded by the phospholipid monolayer, not bilayer, and there is cholesterol also. This is cholesterol. This is phospholipid bilayer. So this chylomicron, phospholipid monolayer. So bilayer, it is present in the plasma membrane structure. It is monolayer in the lipoprotein. So this is phospholipid monolayer along with cholesterol is there. This is cholesterol. Along with this, there is integral protein. There is integral protein. So this is integral protein. So integral protein in the chylomicron is ApoB48, and peripheral protein ApoC. ApoC. This is peripheral protein. So this is the structure of chylomicron. What is the role of chylomicron? Now when there is re-esterification that occurs in the intestinal cell, there is synthesis of lipids, and these lipids along with the protein in the intestine will form the chylomicron. These all structure will form the chylomicron in the intestinal cells. It will form the chylomicron. Along with this, the fat soluble vitamins A, D, E, K, they are taken into the chylomicron. Now through this chylomicron, by the process of exocytosis, this is released into the lymphatics, and through lymphatic it enters the blood. So this, via the chylomicrons, they are taken into lymphatics, lymph, and via lymph, via the thoracic duct, it is into the blood, it is into circulation. The lipids are into the circulation via the thoracic duct. Thoracic duct, it is into the vessels, it is into the blood. So this is how the digestion and absorption of the triacyl glycerol occur. But medium chain, short chain fatty acid glycerol, part of the fat, it can absorb directly, and it can enters into the portal circulation. It can enter into the portal circulation without involvement of chylomicron. So these are the different process. Just wait. See, first word we discussed, emulsification, means the lipids are converted into smaller droplets because the bile salt reduces the surface tension and converts these larger lipid particles into the smaller one. That is known as emulsification. Bile salt, sodium and potassium salt of glycocolate, taurocolate, chinodeoxy means glycochinodeoxycolate, and the taurochinodeoxycolate. These are the bile salt. It reduces the surface tension, converts the lipids into the smaller particle, means it helps in the emulsification, it helps in the mixed micelle formation. Pancreatic lipase specifically cleave the C1 and C3 of the triacyl glycerol and convert triacyl glycerol into monoacyl glycerol and free fatty acids. There is mixed micelle formation, means the bile salt at the periphery and the products of the pancreatic lipase, cholesterol esterase, phospholipase A2 means which products? Monoacyl glycerol, diacyl glycerol, free cholesterol, phospholipid, that is lysophospholipids, it is into the mixed micelle. Okay, they are absorbed into the intestinal cell, the duodenal cell, specifically in the small intestine, that is mainly in the jejunum and ileum. By the process of simple diffusion, these lipids are internalized into the intestinal mucosal cell. Now, in the intestinal mucosal cell, re-esterification occurs and lipid synthesis occurs, and these lipids are incorporated. These means there is problem of carrying these lipids, so they get incorporated into a specialized structure which contain neutral lipids, means the hydrophobic lipids, the amphipathic lipid, and the protein. That structure is known as lipoprotein. So here it is chylomicron, which is important for transport of the dietary triacylglycerol or intestinal triacylglycerol from the intestine through the chylomicron. It is transported to the adipose tissues and the peripheral tissues, means skeletal muscle, heart. Okay, so incorporated into the chylomicron. Chylomicron, by the process of exocytosis, it enters into the lymphatics, and via the lymphatic it enters into the blood. See, so this is a role of bile salt. It helps in the emulsification, it helps in the mixed micelle formation. Now, three important factors are required for the emulsification: bile salt, the amphipathic lipids, and the peristaltic movement, mechanical mixing is required. Okay, so this is larger lipid particles, it is dispersed into smaller droplets, and that's why this action is important to increase the surface area of the lipid particles. Digestion mainly occurs in the intestine by the pancreatic lipase. There is role of cholecystokinin for release of bile and for release of pancreatic enzymes. There is role of secretin for the release of bicarbonate. Short chain and medium chain fatty acid, they are degraded in the stomach by the acid stable or gastric lipase. Pancreatic lipase is the important step. It cleaves the C1 and C3 fatty acid and will form the monoacyl glycerol and the two molecules of free fatty acids. Okay. Now these products, they form the mixed micelle and they are internalized into the intestinal cell by the process of simple diffusion. Then again synthesis occurs in the intestinal mucosal cell and they are incorporated into a specialized structure that is known as chylomicron and carried via the chylomicron from the intestine to the peripheral tissues, mainly the adipose tissue, skeletal muscle, heart, they are carried. So via the lymphatic, they are carried to the blood, and through the lymphatic they are carried to the blood, and this chylomicron is responsible for transport of these lipids from the intestine, means dietary lipid or exogenous lipid from the intestine to the peripheral tissue, specifically adipose tissue, skeletal muscle and heart. Okay. Now the disorders of this digestion and absorption of lipids, that includes mainly the pancreatic insufficiency, pancreatitis, that is inflammation of the pancreas which affect the pancreatic enzymes to release into the intestine. Second, the gallbladder disorders and obstruction to the flow of the bile, that includes the gallbladder stone or CBD, common bile duct stone, that obstruct the release of bile into the intestine. So this also affect the digestion and absorption of the lipids. Then this condition where digestion and absorption of lipid is affected, this condition is known as steatorrhea. Where lipids, large number of lipids, it is excreted in the feces in the steatorrhea, and this is because of pancreatic insufficiency or the gallbladder disorders or various types of jaundice that also affect the lipid absorption. So condition is known as steatorrhea. So this is all about the digestion and absorption of lipid. Thank you.